Hello and welcome to the Figure360 version of Figure 1 for our paper, A Data-Driven Approach to Predicting Successes and Failures of Clinical Trials. This is a schematic for our method Proctor, which looks to address the question of identifying toxic drugs before they reach clinical trials. Drugs can fail for toxicity reasons at any stage of the drug development pipeline, ranging from preclinical work to late-stage trials. Traditionally, this problem has been addressed in preclinical studies, using animal models, or drug likeness methods, which are estimates for characteristics such as how easily a drug can be taken orally. However, when we look at how these methods did on actual clinical trial results, we found that they were unable to tell the difference between toxic and safe drugs. To address this problem, we developed Proctor, our tool for predicting odds of clinical trial outcomes using random forest. The way this works for a given drug, Proctor takes in information about the drug's structure and targets, uses these to derive a set of features that are inputted into a pre-trained random forest model, which outputs the likelihood of toxicity for the drug. I'll begin by breaking down each of the four major feature types into greater detail. We'll start off with structural features of a drug, which include general properties such as polar surface area, as well as more specific details like the number of rotatable bonds. These properties have been integral to existing preclinical toxicity screening methods. For example, in the bottom left, you can see the cumulative distribution function, which shows how toxic drugs in the red have larger polar surface areas than FDA-approved drugs in the blue. The way that we derive these features for a drug, we take the SMILES representation, input that into a computational method, such as ChemMinar, and use that to estimate each of these molecular properties. These properties can then be used to derive another set of features, the drug likeness measures. This includes rule-based methods, such as Lipinski's Rule of 5, as well as the continuous QED metric, which you can see on the bottom right takes on lower values for toxic drugs. Next, we transition over to the target-derived features, starting with expression-based features. The motivation for these features is that the expression of a drug's target in specific tissues, such as the liver, may end up leading to adverse events. For example, in the bottom left, we can see that toxic drugs have higher expression levels in the liver. Since the expression of genes across all tissues are highly correlated, as can be seen in the principal component analysis plot, we decided to collapse all tissue expression data into their first three principal components in order to construct a more robust model. In addition to the target expression patterns, we included other types of properties of a drug's target as model features. These are loss of function frequency and network properties. For example, the network degree of a drug's target is the number of neighbors their gene interacts with in a biological network, as seen in the bottom left. Finally, we integrate each of these feature types together as input into a model that predicts likelihood that a drug would have unmanageable toxicity in clinical trials.